you. So thanks first of all for inviting me and for joining this seminar. And so I am a neuropharmacologist as a background. And today I will try to share with you some methods that we actually try to implement in our daily work to try to investigate more deeply some aspects of the brain neurovasculature on one end, and on the other hand, to try to adapt them in order to investigate the biodistribution of therapeutics once they are administered in the brain. So you will see basically also examples of imaging approaches that, basically based on magnetic resonance imaging, which, by the way, since you are dealing with the possibility of investing on new methods meant for drug development, you must be aware that basically neuroimaging is considered a biomarker from a certain point of view, because definitely you can rely on the results of MRI or specific features that you can highlight by neuroimaging in order to have a so-called let's say, surrogate endpoint, the possibility of having a readout of a therapeutic approach, of a treatment, or to monitor the progression of a disease. So definitely this is an aspect that we should consider and try to consider also on a quantitative manner in order to gain new information. So the way I will address the presentation is basically referring to these papers that we published recently, where you basically can obtain all the images that I will show, as well as the method. So if I, for any reason, skip some details, you will always have the chance to go back and pick up those details there. And there will be some slides in which I actually highlighted this mark, and this means that there will be information that you might want to take notes about, because eventually that might be useful for the final questions eventually. So be careful and take notes at that point. So overall, the presentation will be divided in two parts, basically. So one part mostly dedicated on trying to leverage ex vivo approaches that allows the possibility of correlate fluorescence imaging with in vivo magnetic resonance imaging. And on the other hand, we will move into the possibility of applying some flow cytometry approaches to try to quantify the biodistribution of therapeutics in vivo. And specifically here, the focus is on nanoparticles as drug delivery tools. And the focus will be try to quantify specific internalization in specific cell types. First of all, regarding the first part of the presentation, so I will share with you basically what we did applying this technique, which is called vessel painting. So a strategy to try to visualize the brain neurovasculature, which we couple with tissue clearing approaches in order to have the possibility of investigating the full neurovascular network in a whole mount tissue preparation, to try to correlate them in terms of images with what we had found when we ran a magnetic resonance imaging in vivo. So I will present a few slides on the problem. As usual, if you are in the lab, you know you want to address a specific pathology or investigate specific features of the central nervous system. So you have a problem, a challenge that you want to address. So I will spend a few words on these pathologies, since this was basically the main driver of our research. And then, so once I will introduce glioblastoma, I will show you how we addressed and why we considered important to exploit also at preclinical level magnetic resonance imaging in animal models of disease, and eventually how we combined this analysis with the ex vivo investigation of the neurovascular network through vessel painting technology. The second part, which will be more quick actually, so we will spend some time on showing you basically the way we design a protocol for staining multiple cell types retrieved from the brain, and allow a proper quantification of these cell types by flow cytometry. And here I will share with you some specific, let's say, details and tricks on how to maybe address some possible challenges or what could be the best method in a specific condition. Let's start from the first part. You might be aware that glioblastoma multiforme is considered basically the most aggressive primary central nervous system malignancy. So this is basically also called high-grade glioma. And as you can see from this picture, this is actually real MRI performed on patients. You can highlight definitely what is called a lesion in the brain, which tells you that there is something wrong in the parenchyma. So since I don't know what is the general background of the audience, okay, I will just make sure that you can distinguish what is a T2 weighted imaging versus a T1 weighted plus contrast imaging, okay? So when we run magnetic resonance imaging, we tune the instrument in order to make sure we can highlight the behavior of the hydrogen protons within the water molecules that compose our tissues. And this is a great advantage because basically water composes more than 70% of our body, and we can basically monitor the behavior and also the features. Here we are talking about anatomical features of soft tissues such as the brain. Whenever you will see mentioning a T2 weighted imaging approach, this is a protocol which is very sensitive to the content of water. So the white is, let's say, spots that you see here is basically the water which is in the CSF, which is within the lateral ventricles, basically. On the other hand, when you run a T1 weighted imaging approach, this is very sensitive to the lipid, which is present in the myelin. You see here that basically the space of the ventricles is black, okay? So you have a different readout here. You see brightness, in this case, every time you inject a paramagnetic compound, so a compound which is sensitive to the magnetic field. In this case is gadolinium, a compound which is injected in the blood flow. Usually it is not able to enter the brain, but if it enters the brain, such as in this case, this means that there was a lesion which damaged the blood-brain barrier, typically what happens when you have a tumor, and therefore it extravasated into the parenchyma, therefore you see this brightness. So this is a technique which is diagnostic, but is also useful to monitor eventually the response to a treatment and the progression of the pathology. Nevertheless, the pathology, despite several tests still nowadays, the presence of some therapeutic approaches that you see highlighted here, the pathology is really unfortunately fatal within 5 years from diagnosis. So there is still a lot of research in trying to improve new drug treatments. From my perspective, so we were focused on implementing new biomarkers that could allow better prediction of the prognosis and the response to treatment. And indeed, as you can see from these publications, MRI was actually implemented already in the clinics, and we also exploited it at preclinical level, demonstrating that it was a prognostic marker. So changes in specific features of the MRI run on the brain was capable of predicting the aggressiveness of the pathology over time. So from this perspective, you know, this is definitely an important tool because can help you gaining information in a non-invasive manner. And here you see that according to these publications, changes in MRI could help stratify the patients. So it was basically similar, it's superimposable to sampling the tissue and checking for specific mutations. So it's a very powerful technique, although still there are some limitations and a lot of space for improvement in terms of advancing the accuracy and the sensitivity of the technique. So the open questions from this perspective are mainly due to the intrinsic features or the behavior of the water molecules. I told you that depending on the protocol, you might have edema that might appear. And if you confound edema for tumor, then you might have an issue, a diagnostic issue, and difficulties in monitoring the outcome. So our goal was to set up a preclinical system, preclinical platform that allowed basically to improve the investigation of new MRI protocols, at the same time verifying the accuracy of those protocols by ex vivo analysis. So the model was a standard model, which you might be aware of. So you have an immunocompromised animal. In this case, it was a nude rat. We injected into the striatum different cells which were retrieved from patients. This is a commercial cell line, which is very rapidly growing, whereas this is basically a cancer stem cell line derived from patients, which recapitulates a little bit better actually the features of the tumor that you find in the patient. So we compared two groups of animals, we injected them, and over time we ran progressively magnetic resonance imaging, and the goal was to sacrifice few animals and analyze histologically how the brain looked like, and compare the histology with the in vivo imaging. In these pictures, you can see the in vivo magnetic resonance imaging. On the left, you see the commercial cell line. So basically, the animals in 7 days were needing euthanasia because the growth of the tumor was so fast that eventually you see several highlights here of brightness, which means strong edema, probably also inflammation, that eventually was basically providing a mechanical disruption of the hemisphere where the tumor was present, which eventually required the sacrifice. And you see that basically you can see also here, if you compare the gadolinium here with what is the signal that comes from the T2 weighted imaging, you see a nice difference. And in fact, we measure the volume here, and we calculated the ratio between T2 versus T1. And you see definitely that this protocol is very sensitive to water, so probably edema is giving you a wrong indication of where the tumor is, because indeed the tumor is in this place probably, and you see ex vivo by histology that actually this is the case. So this is the tumor mass. Whereas cancer stem cells, which recapitulate very well the invasiveness of the tumor in vivo, provide a different progression, a more slow progression, and also the signal is less evident in terms of positivity, hyperintensity in the two protocols, as opposed to what you saw with the commercial cell line, and overall the ratio was pretty similar in these two conditions. So starting from this consideration, we said, okay, if you want to improve MRI protocols and we want to do a comparison with the histology ex vivo, we should run the MRI. Here, the values that you retrieve are actual volumes that you measure. Here, you get the tissue, you slice the tissue, and you start measuring areas and quantifying eventually the result. This is time consuming. It might not be very accurate in terms of comparison from a certain point of view. And obviously here you see immunohistochemistry, which I think you know very well as a technology. So you rely on antibodies to stain specific antigens in your tissue. So you need to slice the tissue. If you get good antibodies, you have high specificity of the signal. You can perform it easily on tissue slices. And definitely you can combine very well both the staining for vessels, here CD31 in a very consolidated stain for vessels, with other markers, so you can have multiple parametric data. It is difficult to investigate the vascular network in 3 dimensions, obviously. You can also see here, you see just a fraction of the vessels, because basically you cut the slices, so you damage the tissue eventually. So it's difficult. On the other hand, despite some recent advances in the processing of tissue, it's still difficult to perform it efficiently on wall-mounted preparation. And again, reconstructing the full vascular network becomes complicated, because you will need to merge together multiple sequential slices in order to get the final result. That's where vessel painting came into place. Vessel painting is a technique which actually allows the direct labeling and visualization of the blood vessels in the brain as well as in the other tissues of the body, because basically, so you can refer to these protocols. What you are doing, you can see it here. So you infuse a dye. You rely on these carbocyanine dyes. These are lipophilic dyes, which are solved in a PBS-based buffer. They are injected through intracardiac perfusion. So what you usually do when you want basically to fix the brain in order to analyze it for histology. So before fixing the brain with formaldehyde, you inject this solution which contains a dye like this one, which is based on carbocyanine. During the perfusion, the dye will basically label the membranes in the full vasculature, and eventually when you sacrifice the animal and you get the tissue, you will be able to visualize the full network. So it's easy to stain the vascular network, because as far as you reach all the tissues and the vessels through perfusion, the dye will be there. From a certain point of view, it's cheaper than using the antibodies, specifically if you think that you do not need to stain multiple slices, because basically here all the tissue will be stained. But in any case, if you are not able or you do not want to work on whole mount tissues, you can still slice the tissue and get the signal in 2 dimensions, and that can be measured. Obviously, the problem is if there is any issue in the perfusion, so thrombi or some issues in reaching specific regions, you will not be able to, let's say, label the vasculature in that place. And on the other hand, since this is a lipophilic dye, if you plan to use detergents in your analysis eventually, so you want, for instance, to run an immunohistochemistry on the tissue that you retrieved after this labeling, detergents will basically delipidize the tissue, so you will remove also the tracer. So you must be careful about this and take this into account. So what are these carbocyanine dyes? You see them here. So basically, you have fluorophores that are linked to an aliphatic chain, 2 aliphatic chains. So basically 2 fluorophores which are linked covalently, and both of them are linked to an aliphatic chain. Number here means a different number of carbons. So it might be, such as in this case, di C18 means you have 18 carbons here, so a longer aliphatic chain, a more lipophilic compound, versus di C12 as an example, where you only have 12 carbons in this aliphatic chain, so a compound which is still lipophilic but less as compared to the previous one. These 2 compounds that I show you here have the same spectrum in terms of signal, just different solubility in water. On the left, you see that depending on your final application, you might want to choose a different dye, which allows you basically to stain a different, to stain the same tissue, but with different color. These dyes are, let's say, present in the literature since a long time, because they were originally used as tracers that can be applied as crystals directly on the tissue ex vivo. So you can get cells fixed, tissue which is fixed, and you can still apply the dye. The dye is lipophilic, so it can label the membranes also in the fixed tissues. Okay. In this case, we rely on this property of the dye. We infuse it in living animals, or at least in the when the animals are sacrificed, to allow the possibility to label in vivo before the final fixation. Make sure that you infuse the dye before fixing the tissue. If you do the opposite, you don't get a proper staining. So you first infuse the dye, which means when you usually infuse PBS to remove the blood, there is where you inject also the dye. After injecting the dye, you wash with PBS, and then you infuse PFA to fix the tissue. At that point, you are in good shape. You have your brains, which are labeled with the dye, all the neurovascular will be neurovascular network will be labeled, and then you need to analyze it. You are perfectly aware, I guess, of the very many protocols that have been developed in recent years since the first discovery of the possibility of clearing the tissues. So removing some components, typically in general the lipids, that change the refractive index in a manner that basically allowed you to get this situation. So the microscope light will be able to pass through a wall mount preparation. You see here this interesting publication where several methods have been compared, and depending on the method you use, you can expect different level of compatibility with different techniques in terms of histology, analyzing specific tracers or fluorophores. You might consider two main, let's say, categories in these methods that are passive and methods which are actively, let's say, clearing the tissue. An active method basically already is applied during the perfusion and allow basically to modify the features of the tissue during the fixation. A passive method can also be applied, for instance, on post-mortem tissues. Recently, there are papers in which actually clearing techniques have applied also on post-mortem tissue from humans. So you can basically simply dip the tissue in specific solutions that eventually will remove some features of the tissue which are basically interfering with the light or fluorescence microscopy. From our perspective, our major concern is making sure we are not removing lipids and therefore our dyes. From this perspective, then, we cannot apply any of the methods which use solvents, detergents, SDS, Triton, or whatever can delipidize the tissue. And you might be already aware that these kind of methods are actually the best methods because you get very transparent tissue. The other ones, those that are using these hydrophilic solutions which basically do not contain detergents, are less efficient in clarifying the tissue, but still they can work pretty properly. You have a nice technique in hands, but then you also need to, let's say, take in consideration also some practical aspects. So I have a brain. Think about the brain of the rat, which is pretty big. Let's imagine you can clarify the whole brain. Then you need to visualize it in terms of acquiring images and analyzing terabytes of information in order to reconstruct an image. So this might be something that might be definitely not easy to handle because not all the labs have the proper microscope or the proper, let's say, resources to, let's say, make sure you can handle this kind of information. And this was our case. So how we can somehow get a compromise that in any case will allow us to get a nice and importantly, let's say, efficient and accurate information without affecting, let's say, the quality of our analysis. So we reasoned that when we run MRI imaging, actually, we are not really scanning the full brain at the same time. We visualize the brain by, let's say, virtual slices. So it's like step by step, you scan different regions of the brain, which are close to each other. In this case, the MRI was run by building virtual slices, which were 750 micron thick. So you get the information. So every time you visualize a part of the brain, that is representing 750 micron of thickness. Okay. So we said, what we can do is we know how the brain look like in vivo when we are in the machine. And so these measurements here are basically the physical orientation in the space of the brain of the animal as in respect, let's say, to the floor and the overall Cartesian space, okay? So by trigonometry, we can say if this is my, let's say, my bench, this is the angle in which the brain is sitting on the bench. So after I do the perfusion and I extract the brain, so you see here the brain after the vessel painting. Basically, you can nicely appreciate the vessels, which are nicely stained, at least the biggest one. So what we simply did was, so we just reproduce ex vivo what we saw in vivo. So this is simply agarose gel that has been, let's say, prepared in a manner that we had, first of all, this part of the agarose, which was something like 2%, so quite hard, and it was cut in a manner that it was capable of reproducing exactly the same angle that you see here. You place the brain on top of it, and then you dip the brain into agarose to fix everything. And then you go to a vibratom, and you start cutting slices that are 750 micron thick, okay? So it might look like a naive approach, but just to make sure that was, let's say, accurate enough, what we did was doing a test. So we injected in some sample animal, test animal, some nanoparticles that were paramagnetic. So you have something inside which contains iron. Iron, once you run this kind of MRI imaging, turns out to be black. So you see this is a signal black here. This is the trace of the needle that went down, and the dark that you see here is basically the nanoparticles that were released, okay? We sacrificed the animal and sliced the tissues exactly respecting the previous way of orienting them in space. And you see nicely here that ex vivo in the slice, you can highlight the same signal that you get here, precisely in the same, let's say, spatial orientation as you saw it in MRI. And if you go by histology, Prussian blue staining is basically highlighting the iron, which is contained in nanoparticles that we injected. And I think I can, let's say, show pretty nicely that overall, the signal that you saw here is really coherent with what you observed here. So from this point on, what we did was, okay, let's move on and try to apply this in the context of animals that received the injection of glioblastoma cells, and they were vessel painted. Whenever you run a clearing protocol, for sure, the dimension of the tissue will change. There are protocols that shrink the tissue, other protocols that basically expand the tissue. And if you get an expansion or shrinkage, which is not homogeneous in every direction, at the end of the day, the analysis that you will run will not be really superimposable to what you were expected to having in vivo. So what we did was picking up a protocol which was relying basically on a passive clearing system which was not containing detergent, and we optimized the protocol in a manner that, as you can see here, basically before and after the clearing, we were getting no changes in or at least not appreciable changes in the overall dimensions of the slice. And clearing a slice is definitely easier than clearing the full brain. This is a compromise from a certain point of view, but still it's functional because for us, we just want to compare the accuracy of that specific image that we ran in MRI with exactly the same slice that was present ex vivo. If you see here, these are the protocol in details, which is basically based on fructose and urea to basically, let's say, soften a little bit the tissue. And it turned out that with a slice of 750 micron coming from a rat brain, dipping the slice for 24 hours at 37 degrees in the so-called Fruit 100, which contains fructose 100%, urea 11%, 0% of thioglycerol. This is just meant to counterbalance, let's say, this is a reducing agent, so it wants to reduce the possibility that fructose change in color due to oxidation. This was sufficient to get a good clearing. Here is the simple protocol. So you just dip the brain, you get it clear, and then you get the possibility to go and run the analysis. How do you run the analysis? So we analyzed by standard confocal microscope, so laser scanning confocal microscope with a motorized stage so that we could sample different region and stitch them together to build the full picture eventually. Obviously, you might have some limitations here because you are restricted to the so-called working distance of the objective. So there are, depending on the objective, you might be able to acquire 400 microns or 300 microns, and you cannot go farther than that. Therefore, if you want to scan the full thickness, you must use a multiphoton microscope. And here we were capable of analyzing up to 2 millimeters of thick tissue. This is a little bit some consideration on the timing. So to analyze through a 10x objective a space which is 8 millimeter per 9 millimeter at a thickness of 390 micron, you need between 6 and 8 hours. Depending on the microscope, it might go faster, but keep in mind that you need to take into account also this timing in terms of acquisition, which, by the way, becomes faster if you just apply the same analysis, but you use a two-photon microscope. And this is the outcome. So now you have your in vivo slices, which come from the in vivo MRI. You have perfused the brain, perfused the animal with the dye eye, so you get the vessel painting. You have cut with exactly the same spatial orientation as in vivo, the ex vivo brain through the vibrotome. You clear the tissue, you acquired it with the microscope, and then once you get the image, you can basically superimpose it to what was the in vivo MRI. And as you can see here, what came immediately to clear was that the image that you get in MRI, at least with this protocol, is highlighting a lesion which is definitely not superimposable to the actual presence of the tumor cells. And the main issue here, which is actually proven also from the clinical aspect, is that many cells are actually capable of invading the surrounding tissue, which is the reason why eventually, even if you do chemotherapy and radiotherapy and surgical resection, you will have a reoccurrence due to these remnants around. So this, if you think of an immunotherapy where you have, for instance, an antibody that might be labeled, might be infused, and eventually you cut and image the tissue, you might be able to verify also the specificity of the immunotherapy in reaching the target cells, which in this case were, by, for instance, labeled by GFP to make them easily visible. And so you might eventually screen for new tools that are more selective. Here is another region where you can appreciate very well how all the vascular network is constructed, and this was something that was of interest of us. We were capable of measuring and evaluating also the adherence of the tumor cells to the vessels, which is one of the key aspects of the aggressiveness of the tumor, since this basically induces neoangiogenesis. And this is what you gain from the two-photon microscope. So you see here, this is the z-axis, so you can go in deeper depth within the tissue. So once you reconstruct the network, then you are in a situation where you can really appreciate the full features of the vessels, both within the brain, the tumor core, as well as in the boundary and in the surrounding tissue. You get probably you might consider why I mentioned, let's say, the fact that there are different dyes. In this case, we noticed, so the first analysis were run with dye C18, being more lipophilic, even if you put it in solution and you are very fast in, let's say, injecting the dye in the animal, sometimes it precipitates. So you might end up having issues with the proper, let's say, staining of the tissues. So therefore, our suggestion is to move to the dye C12, which instead is more hydrosoluble, and you can see also here how the staining of the vessels is really appreciable in terms of higher capability of staining the full vessels, suggesting that you reduce the chances that there are some precipitations during the infusion, which might affect eventually the reproducibility of these results. In terms of quantification of the vascular network, you can rely on open source software, you see here, that basically allow you to load the full Z stack of the image that you acquired. Then you prepare your 3D image to be basically, so you first start with the Fiji, so image A, and then you transfer the information that you retrieved from Fiji in terms of multi-TIFF image to the VA3D, which is basically capable of analyzing pretty high dimension pictures. And in this software, you have the possibility of applying this algorithm, which is called MOST, which is capable really of analyzing these vessels and getting a nice reconstruction. This blue image is the way the algorithm basically managed to identify in an automatic manner the vessels, and therefore what you get eventually is a file which is like this, which eventually can be then reloaded into Fiji in this application that is basically simple neurite tracer. So you can find it into the application of Fiji. And this, when you open the SNT, what you will end up having is basically these kind of images where basically the whitish part here is the original image, and all these segments are the way the MOST algorithm basically reconstructed the vessels. Through this information, you get information for each vessels on the length, the starting point, the end point branches, as well as the thickness, which is basically the caliber of each vessel. So you can get quantitative information, and typically the measurements that we ran is vessel density, so the sum up of the volume of all the vessels divided by the volume of the frame of the analysis, the mean radius, so you get the information of basically the caliber of the vessels, as well as tortuosity. This information is critical and is difficult to retrieve if you analyze a 2D image, because in this case, the tortuosity takes into account the capability of tracking the full, let's say, branching of the vessel, taking into account, therefore, the possibility to measure if the vessels are rather straight or capable of do many branches here. In a 2D image, this would not be possible. Another potential application here, in this case, we wanted to track the distribution of cells which were GFP labeled and were infused in the CSF. So these were a very tiny amount of cells. If you want to measure a few days, let's say, after the injection of the cell in the brain, the distribution of these cells by histology, you might want to be very lucky, because if you start slicing the brain, you might miss some cells. Therefore, what we decided to do is we leverage the clearing technique. So we just take a plain slice, 2 millimeter thick, we clarify it, and then we analyze the whole mount image, so making sure that we can pick up also very rare events. The concept of picking up rare events bring me to the last part of the presentation, and tell me if I'm going too long. It will be just a few slides more and then I'm done. Okay. In this case, we were analyzing the biodistribution of nanoparticles. So nanoparticles were labeled with rhodamine. These are polymeric nanoparticles, and then can be exploited to basically release drugs. And recently, we are also improving them in terms of formulation to try to release also genes, so mRNA in vivo. So we injected the nanoparticles in the CSF, and we wanted to see how was the distribution. What we did initially here was just slicing the brain, getting the signal, and then measuring the amount of signal in each region, you see here. But imagine that you want to screen for different formulations and maybe compare different routes of administration. if you want to get at least 3-4 animals per each condition, this will be a huge work and it might be also time consuming. That is where we decided to move from histochemistry, that you know very well what are the pros and cons. In this case, the cons is time consuming and you need to analyze multiple tissue slices in order to get quantifiable measurement. And if you're picking up very rare events, in this case would be the internalization nanoparticles in few cells, it would be difficult to pick up them because you need really a big sampling. That is where flow cytometry came into mind. So the possibility of exploiting antibodies selectively targeted to specific antigens in order to make sure we could analyze properly in a quantitative manner the distribution of the nanoparticles and their internalization in specific cell types. The cons in this case is that you lose the anatomical distribution. So you just get a quantitative measurement on whether my nanoparticle were in specific cell types. I cannot tell in which part of the brain. So that is where you need to make compromises. What we did here is basically, first of all, choosing the right method to get a single cell suspension. So you might be aware that if you want to run flow cytometry, you need to get a single cell suspension. Cells will need to flow through a capillary and then through a flow that eventually will be hit by a laser, and eventually you get a signal step by step from each of the cells that goes in front of the laser. Some typical ways of homogenizing the tissue is using this so-called Downs homogenizer or NICE, which disrupt the tissue in a quite gentle manner, but still will eventually damage neurons and astrocytes. So if you want to analyze myeloid cells, so microglia, for instance, so tiny cells, this might be useful. And it's also useful because it's very rapid. And if you work on ice, you can preserve RNA signatures and metabolic features of your cells, but you lose many cell types. Therefore, we decided to use another method, which basically rather rely on the digestion with a buffer which contains papain, which disrupts the extracellular matrix, plus DNase, which is more gentle and was capable of preserving basically several cell types, ranging from neurons up to oligodendrocytes. In this case, you might consider that since you will see in a moment, you have some incubations. If you are caring about the metabolism of the cells, or let's say the RNA transcriptional profile, which might be affected by these incubation times, you need proper controls, making sure you have methodological controls that allows you to pick up what is really critical features of the tissue. Very quickly, the method starts by mincing the tissue, then digesting first with papain, 15 minutes at 37 degrees. Then you switch the buffer and you basically incubate the tissue in a buffer which contains DNase, so that you basically remove anything that would make it sticky. And then eventually you centrifuge it by using a Percol gradient. So Percol is a solution which has a specific density. So after centrifugation, the debris, myelin debris, and very lipid-containing, let's say, particles will go on the top, and on the bottom you will have your cells. So once you wash these, you retrieve your cells, you are able to stain them with antibodies. And what we chose was a list of antibodies that allowed identification of endothelial cells, leukocytes. This was a vital dye. This was the signal for rhodamine, for the nanoparticles. This is a marker which was developed by Milteni to stain astrocytes. This is useful to stain microglia, macrophages, neurons, and oligodendrocytes. At the end of the day, after you apply this multiparametric imaging, what you end up is a strategy that allows you basically very rapidly, after you sacrifice the mice, in half an hour of incubation plus centrifugation, having the possibility of running into the fax this sample and retrieve information on the presence of microglia, lymphocytes, oligodendrocytes, astrocytes, and neurons and endothelial cells. So if you want to go and interrogate each of these clusters of cells, you might verify, for instance, the percentage of nanoparticle positive cells. And so you might have a possibility to compare different routes of administration or different formulation in a quite easy and fast manner. We are recently applying this also in terms of analysis to verify, for instance, oxidative stress. So you use staining for oxidative stress, and you verify in pathological condition or after specific treatment, which are the cell types which are more sensitive to this treatment. So, very last question, sum up. With vessel painting, we have a strategy that allows us to stain and visualize the vascular network. We can work both on 2D preparation, but definitely this is suitable for a wall mount preparation, provided that you apply a system, a treatment that do not delipidize the tissue. So if you have a passive clearing agent, which does not contain detergent, then you are in a proper situation to basically get your analysis. And if you want to do correlative microscopy, such as in our case, to compare what you see ex vivo from what was the MRI imaging run in vivo, make sure you implement a method that allows you to make sure that you do not change the overall dimensions of the tissue to basically make sure that the comparison is more accurate. And as far as multiparametric flow cytometry is concerned, definitely can be applied on brain single cell suspension. Make sure, obviously, that you run the analysis on freshly isolated tissue. Sometimes people ask me, can I run this on frozen brain? No, there is no possibility, obviously, because once you get your frozen brain and you start digesting it, you will not have single cell suspension anymore. You will have just an homogenate. You cannot run on PFA fixed tissue, although you might think to do the opposite. So you have a freshly isolated brain, you freshly homogenize, you stained with antibodies, and then you fix the staining with PFA. And in this manner, you can run the experiment today, stain your sample, keep them on in the fridge for a couple of days, maximum 3 days, and then go to the flow cytometry afterwards. So in this manner, you can also accommodate the possibility of switching a little bit the timing with the practicality of some experiments. And with this, I will finish, thanking all the people that overall helped me in this journey. And if you have any burning question also after today, feel free to reach out. Apart from this, I'm available for any possible questions and curiosity. Thank you very much for your attention.